All right, we're going to get started with our class, and here's where we're at this week. We're working on teratogens especially now, and fitting those into our course. And we won't have any more class discussions or readings. We'll just have 2 class meetings next week at the end of this semester, and we're working on our capstone paper. Well, continue to add to our journal, and you and I can meet and talk about that anytime. And don't hesitate. You might feel like, oh, you've got a long way to go and things like that, and just let me know, and it'd be great. We could meet. And then, again, it's a pretty open deadline for when you get this done. It's good to let me know, what your plans are, and then when you are done, send me an email, okay? So then, I just have this, because next week we'll try to get back to things like this, of putting together packages for our final exam, and also trying to help organize your capstone projects like that, okay? And so one way to do that is to try to organize this. I've shown this slide several times, and so here I've highlighted as examples of, well, what medicines are known endocrine disruptors, and what would be our best examples of those. And so that's what I think we should be trying to do, is put together those best examples, and have those ready as examples like that. So we had a really great contributions to our discussion on Monday of mechanisms of teratogens, and so I have names up here because on Monday I didn't have a chance to look at most of the contributions before class. So everybody talked a little bit about what they had found, and I want to look back on some of them and try to put them back, put them into our package. And so Ashton brought up methoxyacetic acid, and so it's a artificially synthesized molecule, so built environment and a pollutant and industrial grade solvent or reagent. And I just had never heard of it before, and I looked into it a little bit more and have some more to say about it, but I think it'll fit better if we talk about some of the other examples and then loop back to this one, because there are actually some interesting handles on it, even though it seems like it's one of these examples that has very complicated, well, I don't want to say complicated, they're all complicated, but has many different ways that it impacts health. And so there's that example. And and so I'll get back to this later in class or on Monday, but I think we'll get to it in class like that. So lots of good information about it, and just new to me. So then we spent some time talking about this, what I'll call Accutane, and you're welcome to call it by its more modern name, but Accutane is an easy one to remember. And so it's that prescription acne medication. And so one additional thing that I found that is often the case that we want to be mindful of as a principle is that the molecule that is the drug is not a problem. It gets converted to an active form, and that is both the mechanism for its use as a medicine, but also as its problem as a teratogen. So general principle of all the different teratogens and endocrine disruptors we talked about, several of them are not the problem molecule, but a conversion occurs. And in this case, it occurs in the body. In other cases, we'll see it occurs in the environment. And so that's true for, we'll get to talking about, Madison brought up mercury. And people always say, oh, you mean methyl mercury, because mercury is the elemental form of it, but methyl mercury is the problematic form biologically. And that actually gets converted typically in microorganisms in the environment to methylmercury and then becomes toxic form that are eaten by organisms that go up the food chain, especially as methylmercury. So that's one concept from the Accutane. And then the next thing is we came up with this thing of, wow, you know, it looks like its medical function is to cause apoptosis in glands in the skin that produce oil. And then it can be a teratogen also, and so a sensitive period for the developing fetus in mammals. And we had some hints of that on Monday, is that might be the common mechanism. Common mechanism is inducing apoptosis of cells, both for the medicine and also for the problems as a teratogen. And I looked into that further, and that does appear to be the emerging view, is that. And so it starts to be, Accutane starts to become a pretty significant example for our course. You know, again, gets converted, again, oh, it's a medicine, it's used, and we talked on Monday, talked again about this, that you know, the issue is, how can it be used as a drug? Well, it's used on people who aren't in the sensitive period for the problems that are caused, right? So mom can take the acne medicine, but her unborn fetus would be in sensitive periods to be harmed. And in both cases, for mom and offspring, it looks like the Accutane works by a pathway that includes apoptosis of cells, and that can either decrease acne or disrupt development. And then Luke brought up that its molecular mechanisms of causing apoptosis are well characterized and have come up before in our course, and would be classic examples of what we'd like to prefer to find in our course, which is the molecular mechanisms are well characterized, and you know, if we brought up this as an example, developmental biologists would say, oh, you should look into that. I wonder if this mechanism or this pathway is a factor. So it starts to become a really good example. I think this is the original article that Luke sent or shared with me, and included this figure that we looked at on Monday. And just a restating of that, some of the things we've talked about is, again, about that apoptosis story, and then I brought up this idea of maternal vitamins, and we talked some about vitamin A and beta carotene on Monday, and then I just wanted to type this out, that for a whole bunch of different reasons, Accutane becomes a good example for us to bring to our final exam. It's also a good example for you in helping to guide your work on your capstone project, right? Is to try to not just go a mile wide and inch deep, but actually be able to dive into the different pieces of our course and represent them, and write, you know, for a classmate to remind them of the principles of a course, and then drive into molecular mechanisms and primary research articles about how scientists are learning about these problems and issues like that, right? So again, the idea of it's a prodrug or a pro-molecule precursor that gets converted by some mechanism, either in the host that's affected or another organism. And then this, again, example of this sensitive period, okay? So that's that. And then I just wanted to, since we hadn't talked about this idea before, about this idea of, well, you know, we know about sensitive periods, but then sensitive periods open the door for the idea that, well, that maybe it could be a medicine that's used at other times in the life cycle. And so I just wanted to pull that together again for a kind of, you know, to synthesize some of our learning. So here's just an AI-generated list of examples of medicines that are teratogens. And so that would be, oh, they have a sensitive period for the embryo or the fetus, but not for, we'll say, a teen to an adult in that life cycle. And so we have Accutane, and down below there's that thalidomide, which is in the reading we did for initially as a really famous example from England, where pregnant women would take it as a morning sickness medication, and that disrupted development of their offspring, and they were born with small limbs and other problems like that. So that also is used some now as a medication, but just being careful to avoid you exposing it to developing fetus during the sensitive periods like that. And then another one is this valproic acid, and Bryce brought that up as an example on Monday. So we want to follow up on that because it's just like these others where it's used as a medication in adults, but we have to be really careful not to have a pregnant woman take it because during the sensitive period that valproic acid can cause teratogenicity, can cause birth defects like that. So we have some good examples of those, again, based on this idea of sensitive period means you could use it if it's useful as a drug medication at a different stage in the life cycle. So that's a good thing. And so then I would say, your strategy should be, well, pick one of those and be ready to write about it on the final. Like bring some examples that you have in a package that you have learned more about, not just limiting it to our class discussions or our my presentations, but we're trying to bundle examples together and say, you don't need to learn about all three of those, for example, but learning about one of them would cover all of them as the examples I think we talked about. So that's what you should be doing strategically, is trying to pick and choose from groups of examples, like to start off and say, well, what's a good teratogen that I would want to talk about like that. And we distinguish, teratogens and endocrine disruptors, right? They're overlapping. So what's something that's a good teratogen that fits in? And keep just adding examples of that. I'd really like to write about this. And my goal will be to ask questions on the final exam comparable to the ones that were on the midterm, where we're trying to, let's start talking about some principles we learned about, and then use an example, and you have some examples ready that you can write more about, like that, okay? So that's what we're up to now at this point in our course, is trying to find sets of examples like this, and then say pick and choose of what you want to write about. So to talk about that valproic acid a little bit more, Bryce sent a nice summary, so I just took a screenshot of that, and add this as an attachment when I post the Brightspace recording today, and I can do that for the others as well. And if you ever want anything like that, just let me know, because I can add it to Brightspace like that. Okay, and so a really good focus on early neural development, and so very specific sensitive periods of this to function as a teratogen during development. And so it fits into this, my thing of let's try to put these together right. It fits into this of, well, valproic acid is a good example of something that's used as a medicine, but there's a sensitive period and a very specific times during development in we'll say invertebrates, but in mammals, because it's a medicine, especially, and it would be functioning as a teratogen, causing birth defects as a teratogen. So that's a way to fit some of these examples into that. Kind of this way of trying to build categories and detailed examples within examples in the category like that, okay? And so any one of these three I think would be a good one for you to dive into some, and I'm not going to have a question on the exam that says you must write about Accutane, right? You're going to be able to bring a different example to that question, and so either if you find either of these more appealing, try to learn more about it, but don't feel like you have to cover them all, because I'm not going to say give me the three examples we learned about, because there needs to be more flexibility for you to be able to bring what you learned to the exam, okay? So here's just a contrast with that. So here's this DES, right? And it's a great example for our course. It's an example of medicine, but it's not used anymore. It causes cancer. So, you know, it's just, there's not been a time when people, they found a really good idea where they're like, oh, this is a good medication to use at some other time. It's just not used like that. So there can be good examples, right, of medications that are not used anymore. And so this would be just to be aware of that. And, you know, again, just a great example for driving into our courses like that, okay? And so then, is this a little bit out of place? That's just a little bit out of place. Well, but it goes to this idea that I had, I'll just bring up it again, where I just organized my research, you know, to say, go after, I want to learn about something, and I just said, well, I'll just ask AI about metals, and there are lots of them that are teratogens, cause all kinds of problems. Many different mechanisms are just affected. But what about them? Could they work as endocrine disruptors? Because that's an interesting thing. You know, where we've seen that endocrine signaling requires a bunch of steps along the pathway. And I just thought, you know, that I'd probably be able to underscore for the class that, oh, these metals act at different points on the pathway, maybe on hormone synthesis and so forth. Yeah?
Have we talked about a teratogen that's not an endocrine disruptor?
Did we talk about what?
A teratogen is not an
Have we talked about a teratogen that's not an endocrine disruptor? Yeah, so that's a good question. We have to stop and reflect on that a little bit. And we've talked about a lot of teratogens that cause several problems like that. So any of these metals you would say are, oh, they're a teratogen. And then I said, as a way to try to kind of simplify and dive into some molecular mechanisms, I limited it to say, well, how would they be an endocrine disruptor? Because they cause, and you'll see that'll come up, right? Because I came up with this and I looked and I said, oh my gosh, arsenic causes all kinds of trouble. It's a teratogen like that. And it's not the most prominent mechanism, but I found that it is an endocrine disruptor in addition to, you know, being role as a teratogen in a non-endocrine disrupting way. The endocrine disrupting pathway is a way for us to dive into it. And so there's this caution we have to, you know, always say is for many of these, they are teratogens in addition to maybe being endocrine disruptors like that. But then if we say, have we talked about one that's specifically doesn't have any role in endocrine disruption? It seems that when I look, you know, just about every teratogen are like, yeah, well, here you go, like that. And we'll see, for example, Juliet brought up lead, so a metal, and Madison brought up mercury. And, you know, those just are big time teratogens and they cause a whole bunch of different problems. And you'll see, I dove into those a little bit more because it turns out, well, we'll see, I have some slides about that. But that's a great question. And because we want to be able to write about teratogens and distinguish them from endocrine disruptors. And so I have to think, you know, and I could stand here and think, if we've talked about something that is, you know, not, like, oh, that's not an endocrine disruptor, and at all, like that. And I haven't, you know, I don't know if I've looked at that valproic acid to see if did you find anything of it acting like an endocrine disruptor?
I'm not sure, but you might be right.
Yeah, and so it's good, and that's a good question, and you know, we'll have next week for me to try to dive in and help further organize that. Because another example, but again, you know, like in the reading we've had, we haven't talked in class about flame retardants as big time problems in developmental biology. I just have a suspicion that if I Google are flame retardants endocrine disruptors, they'll be all, yeah, you know, in addition to all the other trouble they cause like that. And So yeah, but that's a great question. That and it's a little bit artificial, right? It's a little bit artificial because, like for example, we'll see. Oh, yeah, lead it can function as an endocrine disruptor, but that's not our exclusive or even primary concern with lead, like that, you know. So part of that is, you know, a kind of artificial, and we got to be careful about that. But it just provides a way for us to organize and dive into some mechanism like that. And you'll see as we talk through today, I think that will help. And so you know, I just done this, you know, as just a way to say, let's dive into some examples. There's a bunch of metals that are teratogens for sure, and what about them? And so Juliet brought up lead, and you know, we learned about lead and its role in replacing calcium. And so I just have a little summary of some of the stuff here. So remember the adverse outcomes pathway. So modern toxicologists would start out saying, well, what is the first thing that lead does that's a problem? And first thing that it does as a problem is it looks a lot to the body. It looks a lot like calcium, and it binds to calcium binding molecules with a higher affinity, and a lot of times really stable. And so Julie brought up, you know, oh, yep, it gets in bones and it stays in bones. And I've looked. I have to try to find some more examples of this idea that you know, that a young girl could get lead exposure that could impact her future offspring because of the idea that it's stored in the bones and it can get pulled out of the bones, as Juliet brought up. And it also binds to zinc sites. And so remember, we have zinc fingers, right? So I said, oh, this arsenic is a great example of endocrine disruptor because it is going to disrupt all the nuclear receptors. So we're going to dive back into that today because it's going to be a common mechanism where, oh, you know, that happens. And again, it's just one mechanism, right? But it is nice to get into one mechanism, and it's not even the main one. And then it also binds to a variety of other protein groups and interferes because of that in so many different ways. But that initial, you know, activating event, initial molecular event is, it looks like important minerals that are used in the body, and if it binds in place of them, they don't function properly, like that. And so widespread problems like that, because those minerals are used in so many proteins in so many different locations. Great, like that. So, you know, a very famous teratogen, and teratogen, and then also very famous in developing children, right? So teratogens are focused on prior to birth, but the brain, nervous system, and other organs still develop after birth. So a great example, but it's true when we try to dive into molecular mechanisms, you're like, whoosh, I'm overwhelmed. You know, like, what could I pick? And so I'd say, well, in some examples, it's good to pick something just to have that, to bring that and say this, like that, okay? So lead, okay? And then mercury. And so another one that's a great teratogen, and Madison sent some notes about that and provided some information about that. I can share that. And I think about mercury as it's an example of a molecule that's going to get converted. So it's not a pro-drug, like we talked about with Accutane, but it is a molecule. When we say mercury, the molecule elemental mercury is not particularly dangerous. It gets converted. And so a famous story about mercury is what's happening to make it dangerous to biological systems. And so here's just a quick image of that from ecologists. So an ecologist would bring up, okay, well, there's mercury moving through the environment. It passes through the environment, it eventually gets cleared out of the environment through some mechanisms. And so the emphasis is that elemental mercury comes from pollution sources a lot, and it gets converted by bacteria into methylmercury and then up the food chain that way. So that would be an example of that. Another example is this idea of what's the molecule that's the problem, and how does it become a molecule of problems. I just ran into this in looking at the mercury some. I can't remember which term. I think we've been using the term bioaccumulation for when molecules accumulate, and I guess there are two terms, biomagnification and bioaccumulation. You can see the distinction there. Bioaccumulation is just they're growing, eating, and it's got chronic exposure. So it's the equivalent of smoking cigarettes all a person's life, right? Is that it accumulates, it just more and more from chronic exposure, right? And so remember, that's a really important concept in toxicity. And then there's the food chain concept, and that's biomagnification. And those are great terms because there is a multiplicative effect, right? In the person fishing, it shows that that person is going to eat a whole bunch of fish. You know, they're going to really magnify the exposure by eating many fish, and that happens all the way up the food chain. So just to clarify those two terms, and because I'm like, oh, I'm not an ecologist, and it's good to keep track of both of them. They're significant in a lot of different settings. This is mercury. And here's just more about mercury as a specific example. So mercury's famous for also, in addition to forming the dangerous forms of mercury, how does it get into the body? How does it get into cells? And that's something that toxicologists really focus on traditionally and currently, is how do we get exposure? How does it get ingested or topical application or inhalation or like that? And then when it's in the system, how does it get into cells? And so that's what's shown here is the specific mechanism for mercury to get into a cell. And again, we don't need to know the details of that, but it's good to be aware of that as one of the principles. If, what's the problem molecule? How does the problem molecule get formed? And then how does it get into cells and into organs to do its issues? So just a little bit more about those, and those are good examples. I think of mercury and lead as being really great, famous teratogens, like Luke said, not really thought of as endocrine disruptors because they just affect so many systems because they're metals and they bind to so many places where a mineral should bind, like that. Or just an atom, sulfur, like that, or minerals like calcium and zinc and so forth, like that. And so but when I look, I see examples of, you can say, is lead an endocrine disruptor? And, oh yeah, sure it is, you bet it is, like that. Is mercury? Yeah. And so I dove into those a little bit since Monday. Prior to that, last week, I talked about, I did that. I said, we'll start specifically looking at metals and their role in endocrine disruption, because I'm like, that'll probably be interesting stuff. And just to, review those slides, oh yeah, this group found that arsenic was an endocrine disruptor, and they had a nice mechanism, a nice assay rather, in lab to test it, because nuclear receptors are transcription factors. And so they had a gene expression assay where they would just activate the gene that codes for luciferase, which is an enzyme that catalyzes production of light in a firefly, and they just in cell lines. And this group was especially interested in amphibian metamorphosis, but their cell line assay used human and rat cells. And the luciferase assay system that's very commonly used to look at regulation gene expression. They were able to do that because they were like, as developmental biologists, we know, these nuclear receptors, they function as transcription factors. So we can use a gene expression assay to test their functionality. And they just took the cell lines and would start adding some estrogen, seeing if that could activate expression of the target gene and cause the cells to light up, and it could. Then they just started adding arsenic, and it came. And then they did it for estrogen and testosterone and corticosteroids, and they said, yeah, we found all the steroid hormones are disrupted. So then here they're looking at thyroid hormone and retinoic acid, because they're all members of the nuclear receptor gene family. in humans, there are 48 different nuclear receptors, and so they're disrupted. And then you're like, oh, how does that happen? You know, how does this metal, right? I can imagine it could disrupt some other step in the endocrine pathway, but how does it disrupt these receptors specifically? And it disrupts them by displacing that zinc. And so zinc is essential in the DNA binding site of all nuclear receptors. And so it just goes back to the same thing, and we'll see that lead and mercury can also do this, and you're like, dang it, you know? And again, not the most common molecular mechanism of action, and we want to really be careful of that, but it brings up an example of just a thing where you're like, oh man, what a classic problem, you know, and just a bummer, right? Because the transcription factor cannot bind to its target genes if it doesn't have a properly folded zinc finger DNA binding domain. So, you know, just like that, and it's just, you know, where we want to get to, where in these examples we want to get to places where you just shake our heads and go, dang it, I can see that, I can see the problem, you know, and so they can function as an endocrine disruptor. Now, we just saw that, you know, for lead, it says, yeah, well, its big deal is it can displace calcium like that. And its next big deal is it can replace zinc, and then it can bind to these sulfhydryl groups, these cysteine thiol groups, and it can serve as an endocrine disruptor, right? And so it's good for us to go there, but also add, you know, cautionary sentences about that. So, you know, I just talked about that and how that's not a surprise, and how all this fits into, you know, an experimental approach. And but, you know, again, for arsenic and these others, that's not the only problem. Arsenic causes lots and lots of problems. Doug Curry studies arsenic and microtubule cytoskeleton of neurons and how it disturbs them. And but it does allow us a way to kind of guide and do some organizing. So again, back to your question of, have we talked about teratogens that are not endocrine disruptors? Well, these metals would be good examples where people would say, like I did, I'm like, I'll be surprised if they're endocrine disruptors, and you look and you're like, yeah, they can do that too, you know? And so, so it's a great question, and brings up, you know, a great theme, principle, concept like that. And so I looked at this cadmium, and this story seems to be the same with cadmium and its mechanism of being an endocrine disruptor. And then, you know, again, binding to that SH group and disrupting the zinc fingers. And so here's just an experiment I showed last week where they have little peptides, and they're just using these little peptides to test the ability of cadmium to displace the zinc, or, you know, displace or substitute for the zinc and cause trouble like that. Yeah, and then Madison on Monday brought up this experiment, and I looked at the research article she brought up, and it wasn't clear from the article. The question I had, was it a one-shot deal, or were they, you know, giving multiple injections? Because Madison used the term that they became resistant, and they were using that term, but does that mean resistant because of chronic exposure? They're developing a kind of insensitivity, and it just, I looked at the article briefly, and it was not clear to distinguish between those two. And but the reason I brought it up is because there are natural proteins in our bodies that are not used to thinking so much about a completely foreign molecule, like maybe lead, but they are built to regulate the availability of the minerals we have in our body, and they're called metallothioneins, and you can see they're just very short proteins that just bind up metals and hold on to them so they don't cause trouble and yet can have that excess amount. And those can be used to secrete from the organism. And so there's some help for exposure to these more xenobiotic elements like lead and mercury, like that. And so just cadmium, and where's cadmium coming from, like that. And so then just back to lead and mercury, and to say what I've already been saying, which is, yes, mercury is known to disrupt the zinc finger motifs of proteins like that, including those found for transcription factors and nuclear receptors like that. So, not its most famous thing, you know, it's interesting because, you know, when we go to my initial look, I said, oh yeah, you know, I doubt these metals will be endocrine receptors, and if they are, we'll be able to highlight other steps than just, you know, being like, oh, they're a ligand that binds in place of estrogen. So, you know, we see that with BPA, that it is an endocrine receptor because it mimics the hormone, and, you know, metals aren't going to do that, but here we find these examples of where the metal can disrupt the functioning of the receptor itself, because it can disrupt all those nuclear receptors. Then, that being said, when you look at lead and mercury too, yes, they can do that. Yes, they can cause trouble with the nuclear receptor. But even when you do that, it shows what I anticipated finding, and that is they cause trouble all along the pathway, from the endocrine gland that's got to make the hormone initially, to the cells that are going to do the responses like that. And so I really expected to find that, and so we want to have that in mind too, right? Is that we can drive in and find a specific molecular mechanism and be like, yeah, that's how they do that. But then when we broaden it out, we say, yeah, and they're endocrine disruptors because they can cause all kinds of trouble along that pathway that we know is there anytime we talk about a hormone. Yep. And exactly the same thing with methylmercury. Yep, they can bind to those sulfhydryl groups, and they can do the same thing, arsenic, cadmium, you know, and so that becomes a great, you know, principal concept of these metals are just replacing minerals that are essential minerals, and they're not the same, and they cause trouble. And they can cause specific trouble, and we have specific examples of that, but then they cause a whole bunch of different types of specific trouble, and as a result can be endocrine disruptors. So that's the take-home messages from, I'll ever talk about that. And you know, it goes back to this slide always where I was just looking for metals that can be endocrine disruptors, and yeah, those all can be, and they can be by disrupting nuclear receptors, but they do other things as well. Yep. And so I think that might be a duplicate slide because it's about lead again, and its actions, and it's not limited to, you know, the nuclear receptor story like that. Yep. Okay, like that. And then, you know, again, this idea that it's mostly the biggest part of the story is it's displacing calcium, and so 90% of the lead in animal is in the bones, but then it's found in soft tissues also, and so this is an example of lead doing something, being an endocrine disruptor without disrupting the zinc fingers, and that is it accumulates pretty high levels in the thyroid. The thyroid is the endocrine gland that synthesizes thyroid hormone, and it disrupts the synthesis of thyroid hormone. So that's called hypothyroidism. A little typo in that word. And it does it by multiple mechanisms. So there are other specific stories that we could turn to say, well, how does lead cause trouble? And even within the role of it as an endocrine disruptor, there are multiple specific stories like that. And so just showing you that thyroid hormone, and I kind of focused on that a little bit because we talked some before about thyroid hormone. Thyroid hormone is involved in amphibian metamorphosis, and so, right, we're focusing on teratogens, right? But endocrine disruptors are not limited to teratogenicity because of post-embryonic development, like metamorphosis, right? And so, we can anticipate that in wildlife, lead is a big problem for amphibians because of their need for thyroid hormone. And the way the thyroid hormone works in amphibian metamorphosis is there is an initial low level of thyroid hormone that's made that stimulates early steps in the frog's transformation, and then the hormone levels go to high levels and high activity, and that causes what they refer to as a metamorphic climax, where the frog body really changes abruptly and it hops out of the water. Yep. And then on my list I had these other heavy metals that are known teratogens. And I looked at them a little bit. It looks like they play roles in endocrine disruption too, like that. I didn't find as immediate examples of them going, oh yeah, this is, something that's, a general principle in endocrinology. But, metals, like that. So lots of stuff about metals. And so then,
when you ask if you were to ask us a question about metals on the exam, would you ask us to talk about something that's not, we don't focus on endocrine disruption? You know what I mean? Or
right. Yeah, kind of like what
Luke
Luke asked about is, what are there teratogens that are not endocrine disruptors, and what do we need to know about examples of molecular pathways? And I'd say that I probably still need to provide an example like that, where I explain in some detail a pathway like maybe the neural tube disruption by valproic acid, where it's really affecting neural development in a different way. Yep. Yeah. And so right now I'd say, well, we don't really have that. We don't really have a mechanism where we, dove into, say, neural signaling and the nervous system and how that can get disturbed by a teratogen and a mechanism of that. So I will be trying to pick up those and fill in those holes next week, I'd say. Yep.
So you might, if we get there, that might be a focus.
Yeah. Because it's good to, expand out with some specific examples that cause trouble other than the endocrine system. Yep.
So when talking about metals, do you think how you might organize a question on the exam would be like question one in our first exam, where you're we talk about all these different things and give an example of how metals can act as teratogens, something like that, or would it have its own focused kind of question?
Yeah, I would say the question in the exam would be it could be about metals, but remember the metals is kind of a made up category that I picked to kind of drive in.
Yeah.
So it might be about metals, but I think it'd be more broadly about pick an example of a teratogen that can function as a endocrine disruptor, and then describe the principles of teratogens and endocrine disruptors, and then explain in detail, what the mechanism is for how that could be caused and what that would affect.
All right, so much broader.
Well, yeah, in a sense, in the question, but then the answer gets to kind of talk about broader principles and terms, and then drive into a specific example. Because we always want to get into mechanisms, because that's the DEVO in the eco-DEVO part of this course, the environmental disruption of developmental mechanisms. So we want to get into developmental mechanisms like that. Yep, okay. And so then back to Ashton's example, and again, it was a new example to me, methoxyacetic acid. And I started learning some about it, and so there's some information about it. And this might be a good example for us to look into. Again, I want to look at it more. This might be a good example of it to look into as an endocrine disruptor, where we could look at its action as being distinct from the hormone receptor. So we see that with these others that, oh, it looks like they could actually, the metal could disrupt those nuclear receptor hormone receptors, but they do lots of other things. And this methoxyacetic acid looks like it has, might be a good example to learn about where it's affecting a specific steps that are different than just being like, oh, and it's messing with the zinc finger domain on the nuclear receptor like that. And so I have a little bit about that. Uh-oh, did I fail to? Oh, this introduces it, and oh shoot, maybe I have failed to include a couple slides. But it just highlights that that it's functioning as a sensitizer. So it's changing at the molecular level, it's changing the sensitivity of the target cell to the hormone. And it's increasing it, but not by binding to the receptor per se, but rather acting on another step in the molecular pathway of the hormone affecting the target cell, right? So we're talking about target cells that would be affected by the hormone, and how do they, what does the hormone receptor cause a response in the target cell? And so in this case, there's it can enhance the activity of nuclear receptors, and so it has effect on reproduction, but not by mimicking the hormone like BPA does, for example, but rather by different steps in the molecular path. So I want to learn about that. Ashton, you do learn any about that initially? I know you confessed on Monday that you hadn't learned very much about some of the stories, but did you run into that idea that the action is by sensitizing tissues to the hormone?
No, I wasn't aware of the androgen and estrogen receptor.
Yeah, so I now look at that because again, we're not, next week is not a discussion, but like we're getting examples of, you're aware that we want to try to pull this together and target it, so you could bring questions and you could bring examples and say, would this be a good example, and things like that. Yep. Yeah, like that. And so I want to, it looks like methoxyacetic acid would be another example where it's a complicated story, multifactorial, multiple problems, causing its teratogenicity, but it might feature a way of another example of endocrine disruption that is a good example for us to pin down. And I have a couple more slides of that, but I just have to glance because I think I got them out of place. Also, yeah. And so I want to learn more about that for next week, because I'll try to find an example of that and also find an example of what Bryce talked about, which is a teratogenicity that really is not an endocrine disruption story. It's targeting another tissue, probably the nervous system and neural signaling like that. Yep. Okay, great. So now the next is to go back to this just general strategy of organizing your learning. And so we've developed some good examples of medicines that can be teratogens, and then we have that built environment. We have good examples of that. So BPA and phthalates are good examples of that. Then we have this pollutants, and the built environment and pollutants kind of merge, right? Because there's some built environment products that become pollutants, like forever chemicals, right? They're part of the built environment, non-sticky surfaces and so forth like that, but then they become pollutants because of their stability in the environment. And so those are kind of together. And then examples of natural sources. And the natural sources that we have so far are limited to, right, my caterpillar is sensitive to endocrine disruption by plants that make that natural hormone. And I think we talked about one other example of that, but I also need to go back and find more examples of that idea of which would be products of evolution. Oh, we have other examples of that where there was that example of the livestock, from our reading, the livestock are feeding on the grasses and there's a weed that they can eat and that will disturb their, oh, and that might be a good example of a teratogen that's not an endocrine disruptor because it disturbs sonic hedgehog signaling in the facial development. So that would be an example. And so we want to pull those together. And I'll try to do that. I'll try to help make a list. And this, I think, is a good way to organize it. Again, it's a little bit an artificial list, but it's a way of organizing our, and putting together our understanding of the situation, right? So, lots and lots of, artificially synthesized molecules, medicines, built environments, or the pollutants that arise from those. And then would be some natural sources. And some of those would be products of natural selection where an organism is trying to defend itself like that, like the plants against the insects. And others, it might just be an arbitrary problem. I think people dispute whether that weed would have evolved that molecule as a defense mechanism or more likely just an unfortunate, a similar unfortunate molecule that causes that trouble. So we want to do that, and we're going to spend next week organizing that, and next week organizing it mostly targeted toward getting ready for our final exam. The final exam date is on our syllabus. Does anybody remember what date it is? In the 6th? Yep. And do you know the time?
11 to 1.
Yep, that probably sounds like it about right. And so I'll get that figured out. So, our final exam will be 2 weeks from today, like that. And we'll be focused on that. We'll be focused on that because I've said, we did that thing for the midterm. That worked out great, but we should just do a little differently for the final where you have to kind of do your best to pull stuff together strategically. And so we'll be spending our week doing that, trying to put these together. And so you're welcome to help on that. And then also, we'll be also welcoming anybody who wants to bring in stuff where they're like, I have a question about my capstone project, because we're all working on that independently. But if you have questions about that, it's likely to be relevant for everybody. Okay, great. So in the context of this, I talked before, and I just have more, more slides about that, about examples. And so we have these great examples. So BPA is just a great example for us in our course as part of the built environment. And it would impact all vertebrates because it's an agonist of estrogen. And so we see fish in nature that are now intersex, they're sterile. Individuals that are neither really a male or female, they have characteristic of both sexes, and it's because of molecules in the environment that are pollutants that can disturb the development of wildlife. And so BPA could be an environment as a pollutant, or it could be in our built environment and something we have in our food container product like that. And then the moleculism, very well known. Good, got it, like that. And another example of that we've been developing is that retinoic acid is a really good one for us to be ready to write about. And again, I think I would try to open the door and have you to pick from it or other examples, but we have some good examples that we have now of retinoic acid functioning that. And then I want to talk some more about phthalates, because phthalates are a very prominent concern in the built environment. And so I want to get some more information about that. Then another big concern in the built environment that I talked about briefly are these PCBs. And so they were banned in, I think it's 1979, but they're essentially forever chemicals in the environment, and so they're just going to be in the environment, and they are big-time teratogens. And they would be, I guess, an example of a teratogen that's not an endocrine disruptor. Their primary mechanism of action is really well characterized, and they bind this protein that's called the aryl hydrocarbon receptor. And the aryl hydrocarbon receptor is not, it's not much of a hormone receptor. And it would have to be a hormone receptor to be calling it an endocrine disruptor. Its function is, as I described before, its function seems to be primarily ready to help detoxify foreign molecules. And so the ligand binding to the site is a kind of a surprisingly big structural shape. And the PCBs are organic compounds that have the 6-ring structure, and they are foreign molecules, xenobiotic molecules, and so they would bind to this. They do bind to this aryl hydrocarbon receptor, but they cause it to misfunction when they do. And so I'll make sure that, I suppose when I Google and say, hey, are PCBs ever endocrine disruptor? I'm like, well, yeah, But I think that, again, that's a great question because we're trying to make this list. And I'll try to produce the list for next week. I'll be like, here's some examples of teratogens like that. And our reading talks about PCBs, and then very closely connected to them are another really famous set of molecules. And so they're products of PCBs and other built environment materials, especially by burning, and they're called dioxins. They also bind to this aryl hydrocarbon receptor. So we should learn some more about the aryl hydrocarbon receptor and what its normal function is and what its abnormal function is when PCBs and dioxins bind to it. And so here's some PCBs, and dioxin is a byproduct of burning of a variety of hydrocarbons. And dioxin is also considered like an old school forever chemicals. It's just really, really stable as a pollutant, right? So examples of the built environment hanging out like forever chemicals. And for the forever chemicals are also on our list. And the aryl hydrocarbon receptor, in order for it to be a detoxifying factor, it's a transcription factor. And so scientists would look at the target genes. And the target genes for it will induce expression of genes that are often enzymes, and enzymes that could potentially detoxify these foreign molecules. So it's really the aryl hydrocarbon receptor is really a way for the body to say, do I have anything that can try to get rid of this? And so the issue is that a variety of xenobiotic molecules like PCBs and dioxins can bind to it, but not allow it to function and try to detoxify them. And so it's a good example of, I'd say these are good examples of primarily teratogens, and probably a pretty good primary main mechanism of action. And so this cytochrome P450s are just enzymes that are metabolic enzymes that are used to digest complex molecules, and maybe some of them could digest this odd molecule that the body's exposed to. And there would be reasons for the aryl hydrocarbon receptor to evolve before humans started producing PCBs and dioxins and built environment molecules and pollutants, because of the idea that organisms could be exposed to a foreign molecule that's not part of their natural metabolic system of digesting, and have to be able to try to induce a response. Lots of this happens in the liver. The liver is considered a detoxifying organ and has this inducibility with of these enzymes with the aryl hydrocarbon receptor. So that's a good mechanism, And I think probably, Luke asked that question, I was thinking, but I think probably these are gonna be good examples of teratogens. PCBs and dioxins, good examples of big time teratogens that, again, even if I find they have some endocrine disrupting function, I would probably still lean on saying, yeah, those are teratogens that are not specifically endocrine disruptors, and they work by disturbing this effort to try to detoxify, try to eliminate them from the system. Okay, and so in that aryl hydrocarbon receptor story, in developmental biology and cell molecular biology, we talk about, well, the transcription factor, and that transcription factor is going to regulate a set of genes. So transcription factors bind to the regulatory regions of genes and either activate them or repress them. And in those fields, we would call those the target genes. So just to provide an example of that, so in our bodies, when we were children, we had all the genes needed to make a human and live a human life. And that included the genes that need to get activated during adolescence for us to mature, right? And another example would be in an insect. In the caterpillars I study, the larvae I study, they have all the genes to make them all. They're just not using them, okay? So the hormones, estrogen and testosterone in vertebrates and disteroid in insects that cause sexual maturation, those hormones must activate a bunch of genes. Sometimes those hormones and their receptors activate genes that are other transcription factors. So the receptor doesn't need to directly activate all the genes that are used during adolescence that haven't been used yet in the life cycle, but they activate a lot. And those are referred to as the target genes. So people talk about the estrogen receptor target gene or estrogen target genes like that. And so that's what's being talked about here. And so this is the aryl hydrocarbon receptor, and they're talking about these gene battery. And that's just this thing where there's a classic set of target genes that when the aryl hydrocarbon receptor binds to a xenobiotic molecule, it is going to increase the expression of those. And all of these are enzymes that are involved in metabolism. And and all of us have learned something about metabolism and glycolytic pathway and the Krebs cycle and stuff like that. There are really complicated metabolic steps in our bodies, and these are some enzymes that perform some really kind of surprising enzymatic reactions. And so this just brings up the idea that the aryl hydrocarbon receptor as a transcription factor has a function, and its function is to activate expression of a set of target genes. And the famous ones are enzymes. These are, again, normally enzymes that are found in the liver. They are found in all the cells, because the genes are in all the cells, but mostly people think about the liver when they think about the activation of these. So again, that's its job. And so, it's, I think, well, I know we wouldn't call the aryl hydrocarbon receptor an endocrine, endocrine hormone. It's not binding to hormones primarily, like that. So it has this set of genes. Then, so that's good, just to be good to have that example. Then it turns out that the aryl hydrocarbon receptor has some additional roles like that. So it plays a role in immune system activation. So these PCBs and dioxins, they cause trouble, and it's kind of a two-step problem. One is you aren't going to be able to activate an effort to metabolize us. And then two, we're in your body now, and you can't get rid of us, so we're going to be chronic, right? And so these forever chemicals are concerned because they're going to be in nature forever, we'll say, really, very long time. Organisms can't break them down. And they're going to be in your body for a very long time, also like that. So it's this two-step thing that becomes a satisfying example where you're like, dang it, we blocked any ability to try to detoxify, try to destroy these molecules in the liver, and then there they are, and the aryl hydrocarbon receptor plays a role in the immune system. And so we're going to also block your ability to do that. So we're hanging around and we're going to cause trouble. And so a famous thing for the PCBs and dioxins is immunosuppression, like that. So reducing the ability of the immune system to fight off infections like that of any kind. So that's a classic problem with these chemicals like that, right? There's that just image again, and then they bioaccumulate, they biomagnify, right? These would fit in, as would any of those forever chemicals, into those concepts, right? In nature of, accumulate over the lifespan or magnify up the food chain, like that. Yep. So yeah, Luke, I'd say I'll look into this, but I'd say I'll come back next week and be like, yeah, well, there's some good examples on our list that you asked about. And then these forever chemicals, right? They cause toxicity, a variety of different types of toxicity, including their teratogens. And so there are a variety of abbreviations, PFOS and so forth, that are out there on products now, and they include toxicity issues that are them functioning as teratogens, like that. And if you Google it, you can find that they can play a role as endocrine disruptors. But again, I think for some of these, we'll start trying to lean towards saying we'll emphasize them as teratogens and not being like, oh, if you ask, are they? Because they probably are, like that, okay? And so there's just examples of that. I forgot that I'd already just glanced at that for some of these molecules that are on products nowadays, like that. And so that's that, and we'll try to add to it. And and then after that, I just have some more slides like that. So we've got a few more minutes, but I'm actually realizing that I think we're in a good place to stop today, like that. Do people have questions or issues, anything like that, about anything at all? And so you don't have to chase down methoxyacetic acid further, but you can try to bring anything more. The questions are great, and again, next week, our next 2 meetings will really be leaning toward organizing our learning to bring it to the final exam, but also constantly kind of mirroring our work on, finishing up our projects. So I think we're good for now. Yep, Bryce?
How much of the, the final's more focused on what we've learned in the second half, because we haven't had an exam on it, right?
Yeah, I think I said this before, that the final will probably have one question about the first half.
Oh, okay, cool.
Yeah,
Perfect.
and because the final, it does cover our whole course, but, we had an exam for the first half already. And so I would say that it'll have one question. And it'll probably have 6 questions on the final. So 5 on the second half. And, yep.
Will the one about the first half be more general?
Well, I think, remember when I was a student, I think that's just such a natural notion. But I think that the answer, I'll hope, will be that, you bring specific knowledge to the answer. But it'll be like on the first exam where I try to craft questions where I'm like, a broad question that opens a door. So, I think for studying for the final, it would be good to go back to a classic example that you, even, I think, looking back at your first exam, you might end up bringing some of that same thing to the question, like that. Another thing would be to say, to compare and contrast the first half with the second half, right? Because it could be a question framed like that. Like what were the features that distinguished the first half from the second half? Because there were some similarities and some differences, like that. And so I think it would be good to review your first exam and notes that you took for preparing for the first exam, and then be ready with specific example or two, you know, that you could dive into, you know. So in that sense, you know, yeah, general, but expecting that in the answer, the student brings specific details. And always remembering, we really like the thing of saying, framing the situation and diving into developmental mechanisms and cellular and molecular mechanisms like that, and the ones we ran into, right, like that. So, like a quick example would be, you know, compare and contrast what we learned in first half and second half, and you were like, well, we learned a bunch about hormones on both, right? Hormones are really important in development, and they're really vulnerable for disruption, and they're also really great opportunities for phenotypic plasticity, you know? And you just slam dunk the exam on my desk and walk out into the sunshine. Yeah, let me just tell you this, because it's a great story. You ready? So, this is a great story, and it features Juliet, because I'm just remembering it about the final exam. So, Juliet took developmental biology like a couple years ago, right? And I teach it in the fall, and so cloudy December like that, right? So, years ago, I had a student named Julie, and you don't know Julie, and the developmental biology class was in this classroom like this, and it was like a great day, and the students were taking their final exam, you know, and I look up, and there's just a classic sunbeam coming in the window, and it's on Julie's hands and exam. I'm just like, my God. And it's like the rest of the classroom is dark, and it was like a biblical thing going on. No, this is great. And so I said out loud, oh my God, everyone, I'm sorry to interrupt your exam, but there's obviously some sort of celestial power coming in and really impacting that student's exam, okay? Right? When you took your developmental biology exam, the exact same thing happened. I was like, this is Julie, and now it's Juliet, but yeah, it wasn't quite as dramatic. And I learned from that Julie that it was not cool to bring that up. Like she said, like that was not helpful at all, you know? You know? It's just